everyone. I'm glad to introduce my friend and colleague, Jenny Zambrono, who is a forest ecologist and also currently an assistant professor at Washington State University. She did her bachelor's degree at the Universidad of Los Andes in Colombia, where we briefly overlap, and after that she did her PhD at the University of Illinois at Chicago. Then she went to the National Social Environmental Synthesis Center as a postdoctoral fellow, and there we also overlap because her mentor at the time was my PhD advisor, Nathan Swenson. And today she's going to present some of her, thank you, most recent research. So I'll let you with Jenny. Thank you.
Yeah, thank you for the introduction and thank you for the invitation. Today I'm going to talk about the use of, can you hear me well? Yeah. Hello? No? Not yet?
Yeah. Let me see. Try now.
Can you hear me? Better? Okay. So today I'm going to talk about the use of trait-based approaches to understand the drivers of forest dynamics. My main research interest is to understand the drivers that are shaping forest systems. But why do I study forest?
I think this is going to be better.
But then it's going to be like in between this and that.
Okay.
And then
Can anybody hear her right now?
no.
No?
Okay.
All right. Try now.
Okay. There we go.
Sorry. Thank you.
Okay. So let me go back. So my primary research interest is to understand the drivers that are shaping forest systems. But again, why do I study forest? Forests provide key ecosystem services, such as the provisioning of clean water, food, and fuel. They also regulate important natural processes, such as plant pollination, climate. They offer other types of benefits, such as cultural and recreational. And finally, they support important functions, such as nutrient cycling and soil formation. So as I mentioned, today I'm going to talk about how I have used these approaches, or trait-based approaches, in my research to address important ecological questions. So these approaches use information on functional traits that are physiological, morphological, and phenological features that are influencing the survival, growth, and reproduction of individuals. So they are determining species interactions with the environment, and they're critical to understand the functioning and stability of an ecosystem. So some of the examples that I have here is seed mass that is related to reproductive investment. We have leaf area that is associated with resource acquisitions. And finally, we have here wood density that is associated with plant support and water storage. So for the rest of my talk, I'm going to present some of the projects that have been involved using these approaches. I have used them to understand the drivers that are shaping forest systems, mostly in the temperate forest, but also some of the tropical forests. And also, I'm interested in using them to understand how forests are responding to changing environments, especially in the context of forest fragmentation. So part of my research explored the role of species interactions in structuring forest systems. So we have that individuals might have these positive or negative interactions with close neighbors. So we have that an increase in neighborhood density can lead to a strong competition for resources. So in other words, we have that individuals that are in these crowded neighborhoods are going to experience like a stronger competition for the same resources, and they might also have a decrease in performance. While individuals that are growing in less crowded neighborhoods, they not experience that strong competition, and they might have or experience an increase in survival and growth. So these density-dependent effects have been suggested as an important force that are shaping forest systems, and also plant communities in general. But these effects tend to vary with climate. Previous studies have described a strong interaction between the abundance of individuals and climate. So we have that, or is expected that toward the edges of these climatic gradients, a decrease in the abundance of individuals. So if you think about very high temperatures or very low temperatures, I'm going to limit the amount of individuals that can establish in these areas, right? What we have in these optimal areas, we can expect to have an increase in the survival and growth of individuals, an increase in performance, but also because the climatic conditions are better in these areas, we can also expect an increase in habitat, in abundance of individuals, and therefore an increase in competition. So to explore some of these ideas, I conducted a tree demographic study at a continental scale. This was in collaboration with Nathan Swenson from the University of Maryland, that was my postdoctoral mentor, and also Philippe Marchand from the Université du Québec. So we hypothesized that tree performance in terms of the survival and growth would be affected by increased neighborhood crowding in suitable habitats, or as the environmental conditions were better. And also, we hypothesized a strong interaction between those neighborhood effects and the regional climate. So we used 14 years of tree survival and growth data that were obtained from the forest inventory analysis, and we included forests from the northeast of the US, and also forests from Puerto Rico, over here. The climate effects were modeled using a habitat suitability index that was determined by matching the vegetation data with climate maps that were downloaded from BioClim. So if you want to think about the suitability of the habitat was defined in terms of temperature and precipitation data. And this index, really what it does is allow us to understand how the suitability of the habitat might be affecting the species interactions. We also use a neighborhood model, and this was to determine what would be the effects of close neighbors on the survival and growth of a focal tree. So the neighborhood was determined by using a fixed radius. In this case, was just the size of the plot, but it has been used before with 20 meters, depending, it varies with the forest study. So what we did is we determined the distance between the close neighbors and the focal tree, and also the size of all those neighbors that were located within the neighborhood. So using this information, we calculated a neighborhood crowding index to understand those competition effects. And that was varying as a direct impact of the size of the neighbors and an inverse function of distance to neighbors. So if you think about this index, it's telling us how crowded is the neighborhood of the focal tree. So can we expect to see an increase in the performance or survival growth in terms of the neighborhood effects? But also, we were interested in understanding how the traits of these close neighbors would be affecting the performance of those focal trees, and also how these effects would be varying as the neighborhoods become more crowded or less crowded. So an increase in neighborhood crowding might lead to an asymmetric competition that is leading to these trait hierarchies, or what we call this hierarchical competition. So what we have here is that individuals that have more suitable traits are going to be better competitors compared to individuals that have more unsuitable traits. That may be experience an increase in the decrease in the survival and growth because higher competition of resources as these neighborhoods become more crowded. So as an example, we have that taller individuals might be better competitors. They're getting more light, more resources than smaller individuals. And therefore, they're experiencing weaker competition as these neighborhoods become more crowded. So using this information, we calculated a crowding index that was dependent on these trait hierarchies. So here we have what I explained before, the distance and size of the neighbors. But we included this difference of the trait values of the focal trees and all the close neighbors. In addition, we might have also that individuals that are growing in more diverse neighborhoods are going to experience weaker competition compared to individuals that are in these less diverse neighborhoods. So these individuals that are located here are going to experience, or we're expected to see a decrease in the survival and growth as competition has become more stronger for resources. And those effects are going to be exacerbated as the neighborhoods become more crowded. So in other words, functionally similar species or species that share similar resources are going to expect it to have this increase in competition. And this is going to be worse if the neighborhoods are more crowded. So similar to hierarchical competition, we calculated this crowding index that was dependent on these niche differences. And in this case, we just took the absolute differences between like the trait values of the focal trees and close neighbors. We use traits that are known to be related to the survival and growth of plants, and also that play key functions. And these included seed mass, as I mentioned before. This is related to the reproductive investment, but it also tells you about different strategies in terms of resource acquisition. We have maximum height that is related to light interception. Wood density, as I mentioned before, is related to plant support and defense. And we also included like several different leaf traits that included the leaf area, a specific leaf area, leaf carbon, nitrogen, and phosphorus content. And all these traits are related to resource acquisition and growth. So now we get to the fun part, the models. So we use hierarchical neighborhood models. We were using information at the species level, which was like the trait information, and also at the individual level, which was these effects of like when neighborhoods became more crowded or less crowded. So we model survival and growth as a linear predictors, and we constructed separate neighborhoods models for each of the functional traits that were included in the study. We included a climate effect just to understand how the suitability of the habitat was impacting the survival and growth of the focal trees. We also included a trait-climate interaction, so how the trait effects of the focal trees would be mediating the effects or the responses to an increase in the suitability or decrease of the suitability of the habitat. And finally, we also included this climate-neighborhood interaction. So with this, we wanted to better understand the role of niche differences and also hierarchical competition, and how these effects will be varying with the neighborhood and also mediating the response to climate. So today, I'm just going to focus on the results related to hierarchical competition and niche differentiation, but we also found other effects that were significant. For the graph, what we have is like the circles represent the posterior mean, and the lines represent the 95% confidence intervals. If you see, those dark circles are the ones that represent significant effects. So what did we find? We found that there was an evidence that hierarchical competition was affecting the survival of trees in the Northeast. So what we have is that as crowding conditions increase because of the suitability of the habitat, we have the individuals that have higher specific leaf area values. So what does that mean? They have higher photosynthetic capacity, have better survival. All right, so this was just for the hierarchical competition. We also find evidence that niche differentiation was affecting the survival of trees in Puerto Rico. So if you see here, what we have is as crowding conditions increase, and here is like the more diverse and less diverse neighborhood. So as crowding conditions increase, individuals that were growing in more diverse neighborhoods in terms of seed mass had better survival. So individuals that were located in neighborhoods that had a mix of species with small seeds and large seeds had better survival than individuals growing in less diverse neighborhoods. And this makes sense. I mean, they were showing like different resource acquisition strategies, right? So they were not competing for the same resources. But then, for the northeast of the US, we found an opposite result. So again, we found evidence of niche differentiation, and in this case, it was affecting the growth of trees in the northeast. But in this case, individuals that were growing in more similar neighborhoods in terms of wood density were growing faster. So individuals that were located in neighborhoods that were mostly composed of species with light wood, they tended to grow faster than individuals that were located in more diverse neighborhoods. So light wooded and also dense wooded. Again, this makes sense. I mean, light wooded species, what they're doing is that they are allocating more resources to vertical growth, and they're not allocating resources to plant defense, for example. So they tend to grow faster. So if we go back to the hypothesis, for the first one, we found evidence that there was these strong density-dependent effects that was promoting the coexistence of functionally similar species in the Northeast, but we found the opposite in Puerto Rico, and that was a very interesting result. In addition, we found that there was a competition for limiting resources. So when I talk about resources, light, space, nutrients, and in this case, this competition was leading to strong hierarchical competition and niche differentiation that was affecting both systems, temperate and also tropical areas. But I think the most important result was like this strong interaction that we found between the local neighborhood and climate. And this highlights the need for future studies to include that interaction that is often omitted, but it's critical to understand the dynamics of this forest. But these functional trade approaches that I just described is not without limitation. I mean, some of the functions are more difficult to assess with these approaches. And so, for example, plant defense is one of those examples, and it has been ignored in community ecology because it's not easy to take that function in the field. But using like recent advances in bioinformatics, such as transcriptomics or genomic analysis, it offers an alternative to understand these key functions or the role that they have in community ecology. And I'm going to show you a study that was part of this special feature that was published in 2017. So the role of plant defense, it tends to be underscored in community ecology, even though it has long been recognized the importance of plant defense as a driving community dynamics. When it was in the early 70s when Dan Jansen and Joseph Connell independently proposed that explanation of why we observe this high diversity in tropical areas. And that became the Jansen and Connell hypothesis. So what they suggested is that these areas that are near the parental tree are going to experience a higher number of seeds and seedlings, and therefore also higher number of attacks by natural enemies. And when I talk about natural enemies, it could be pathogens, herbivores, pests. And so what happens is that these areas become inhospitable for the seeds and seedlings compared to areas that are far away from the parental tree. So by doing this increasing attacks of natural enemies, we also have these areas allows the coexistence of different species, and therefore that was the explanation of higher diversity in the tropics. So in other words, if we have that individuals that are occurring in high conspecific density, or individuals are from the same species and sharing the same natural enemies, we could hope, or we can expect to see like a lower survival and growth compared to individuals that are growing these heterospecific density neighborhoods where individuals are not from the same species and therefore they're not sharing the same natural enemies. So to explore this idea, we conducted a community transcriptomic analysis in collaboration with Nathan Swenson. This analysis, or this study, was conducted at the Wabicon Lake Forest Plot, which is located in Wisconsin. And seeds for 21 species were germinated in 2010. Then the subsequent seedlings were collected for RNA extraction and sequencing. And from those, we identified 27 homologous genes that were related to plant defense. So the idea with this was to see if conspecific density was leading to a negative effect on the survival and growth of focal trees. We expected also that heterospecific density will have a positive effect compared to the conspecific density. But using that information on plant defense, we were also looking at if the neighborhood dissimilarity in terms of those plant defense genes would be associated with higher performance. So we use a similar approach, these hierarchical models, again, information at the species level, which was the gene defense, and also we included individual level information, again, that was like related to the neighborhood crowding that you see here. We constructed separate models for the 27 homologous gene, and again, survival and growth was modeled as a linear predictor. We included this gene effect, but also we were interested, again, in looking at the conspecific density. So what happens if an individual is growing in a neighborhood where you have all the individuals that are from the same species and sharing the same natural enemies? And also a retrospecific density effect. So now, what happens if the individuals that you're growing with are from different species? And finally, we included this neighborhood dissimilarity effect. So we wanted to see the effects when the neighborhoods shared the same plant defense genes. All right, let's get to the fun part, the results. So we found that when we're looking at conspecific density and tree survival, as we were expecting, there was a negative effect of conspecific density dependence, but this is not something new. I mean, there's growing evidence that these effects are shaping forests as well in the tropics and also in the temperate forests. But I thought there has been some debate lately on how we are measuring conspecific density and therefore the role of these effects on plant communities. As we expected, we found a positive effect of heterospecific density in tree survival, but this was a weak positive effect, as you see here, compared to what we observed with the conspecific density. And similarly, as we were expecting, we saw this positive effect of neighborhood dissimilarity on tree survival, but again, this was weak. But what this was telling us is that individuals that were growing in more diverse neighborhoods in terms of plant defense had higher survival. So that's what we were expecting. Now, when we look at tree growth, similar to survival, again, we observe this strong effect of conspecific density, but contrary to what we found in the survival, now we see that heterospecific density was negatively related to tree growth. Similar to survival, we observed that individuals that were growing in more diverse neighborhoods in terms of plant defense tended to have higher growth. So what does this mean, all this? If we go back to the hypothesis, as we were expecting, there was this strong conspecific negative density-dependent effects that was reducing tree performance, but we found mixed results when we look at heterospecific density. But I think the most important thing was that, as we were expecting, we found that tree performance increased in more diverse neighborhoods, and this is consistent with the Jansen and Kono hypothesis, right? So if you're located in a neighborhood that they don't share the similar strategies, you tend to do good. And in this case, it was plant defense. So I think, going back to this hypothesis, and this being one of the first studies to link transcriptomic data with community ecology, I think this approach offers a great opportunity to look at these functions that they tend to be omitted because they're more difficult to measure in the field, but are necessary to understand these forest dynamics. So now let's change a little bit. So I've talked about how I have used these approaches to understand the drivers of that are shaping the forest systems. But for the second part, I'm going to talk about how I have used those to understand how forests are responding to changing environments. And as I mentioned before, in terms of forest fragmentation. So forests around the globe are experiencing unprecedented rates of deforestation. We have seen this, especially in tropical areas. We have seen this effect. And this is threatening biodiversity and also adding an uncertainty of how those forests are going to respond to future changes. According to data from the Global Forest Watch in 2017, we lost an area of 15.8 million hectares. If you don't know, this is equal to losing 40 football fields of trees every minute for an entire year. And let me tell you, this is not soccer fields, right? This is American football. So just imagine that. But unfortunately, forest loss almost often leads to forest fragmentation, or this degradation of a continuous forest into several small isolated patches over time. The problem with this is that it interrupts critical ecological processes, so it could be interrupting the plant pollination, seed predation, seed dispersal, or that movement of the seed away from the parental plant, and also herbivory. And the problem with this is that it reduces the number and also the diversity of species. And we can expect that it will have also a negative impact on the functioning and stability of the ecosystem. So forest fragmentation is a very complex problem, and it requires a multi-scale approach to link processes that are occurring at the landscape level with processes that are occurring at the fragment level. So we have, as habitat loss occurs, we have the fragmentation of the forest, But the thing is that as time continues or progresses, we see that those fragments tend to become more isolated, and also that matrix or the area that is surrounding those fragments tend to become more distinct than the original forest. So in that matrix, we might find often like the presence of pastures or some cultivations too. And at the fragment level, what happens is that we have that the fragments become smaller in area, but also the complexity of those fragments tends to be reduced as fragmentation progresses. Another thing on top of that, we have these edge effects. So fragmentation often leads to the creation of these edge habitats that we see here, the light green. And the problem with these habitats, what we observe is that there is a change in abiotic conditions. So we tend to see an increase in the temperature, increase in wind turbulence, and therefore an increase in desiccation. And that might have a negative effect on the survival and growth or population dynamics of the individuals that are in those habitats. So in a synthesis that we published last year in Ecology, we were looking at the effects of both habitat loss and fragmentation on traits that are related to important aspects of the species life history. So we have the dispersal, establishment, and persistence. So what happens is that fragmentation might have a negative impact on the traits related to dispersal or that colonization ability of the species to go to these newly formed fragments. The problem with this is like this might lead to a genetic deterioration, so a loss of genetic diversity, and also an increase in inbreeding. In addition, we might have a negative effect on the establishment or that tolerance to novel abiotic and biotic conditions. So we can expect to see this decrease in survival and growth, even reproduction, and also these altered species interactions. You might see a decrease in plant pollination and seed dispersal too. And finally, we might see also these negative effects on persistence, so that avoidance of small populations. And the problem what we have is that with very small populations, you have an increase in the extinction rates and genetic drift, as well as a decrease in reproduction. So when we were looking in the synthesis, or the studies that we include in the synthesis, they yield mixed results. So we can see that the functional diversity or the diversity of those traits that are on those fragments, it was like positively or negatively related to fragmentation. And so that raises the question whether that lack of consensus is like the result of not including these indirect effects. So the continuous arrows, those are like the direct effects. So we have the matrix quality, edge effects, all these fragment processes or habitat loss that they have a direct impact on those aspects of the species life histories, right? So dispersal, establishment, and persistence. But the problem, and that's why I was saying it's such a complex problem, is that we have also these indirect effects, and they tend to be forgotten when we're looking at the effects of fragmentation. So to look at these ideas, or explore these ideas, I conducted a study that is currently in review in +1, and this was in collaboration with Noel Beckman from Utah State University and Novi Cordero from the Chicago Field Museum. So we're trying to see the effects of forest fragmentation on functional diversity. And this study was conducted at the East Usambara Mountains, which is located in the Amani Natural Reserve, and this is in Tanzania. So really what we're trying to see is that if the variation in functional diversity was the result of these fragment and landscape level processes. So we included functional trait data again. We included like seed length that I explained before that is related to this reproductive investment, wood density, maximum tree height. But also we were interested in this dispersal mode. So whether the seeds were dispersed by animals or by wind, because that might be also like related to the degree of isolation. And also shade tolerance, so whether the species occur early in the succession and they were light demanding species, or they occur late in the succession and they were shade tolerant. And also, I use a structural equation model approach to investigate both the direct and indirect effects of landscape level and fragment level processes. So we have the direct effects for the landscape level. Metrics quality and isolation can have a direct effect on functional diversity. And at the fragment level, we have fragment area, shade complexity, and distance to edge that we use as a proxy of the amount of edge habitat. But we also included this indirect effect. So matrix quality can exacerbate the effects of isolation, as well as exacerbate the effects of this amount of edge habitat or edge effects. In addition, we have that very small fragments can also lead to an increase in the edge habitat and also lead to a decrease in the complexity or the shape complexity of those fragments. And finally, we also included this indirect effect of shape complexity on distance to edge. So as you have a reduction in the shape complexity, you also expect to see an increase in the amount of edge habitat. So we define functional diversity in terms of functional richness, eveness, and divergence. And functional richness is just the amount of that functional space that is occupied in the community. I won't go into a lot of details about these metrics. But pretty much, it's telling you about different aspects that you could expect of those traits that are present in the community. We also included functional eveness, that is just that regularity on the distribution of those traits. And finally, we included this functional divergence, which is just that variance of the trait distribution in those communities. All right, so what do we find? So we were expecting, we found these strong effects that matrix quality was exacerbating the effects of isolation, but then we didn't find a direct effect of isolation on functional richness. But what we did find is that distance to edge had a negative impact in functional richness. So a subsequent analysis using just community-weighted means, what we found is that those edge habitats was mostly composed of species that have light wood. But then we look also at these indirect effects, and what we found is that these edge effects were also mediated by how big or small was the fragment. So very small fragments will have larger amount of edge habitat. And the problem with this is that it was exacerbating these effects of distance to edge. And on top of that, we have that as we have a reduction in the complexity of the shape, we also have like higher amount of edge or bigger edge habitat. And that was also resulting in a higher impact on the functional richness. So this was for functional richness. For functional eveness, we found a similar pattern. But in this case, isolation had a direct effect on functional eveness, and it was decreasing. So very isolated fragments tend to have more species, like wood species, but also species that were dispersed by wind. And the problem with this is that if you think about, as you increase the distance between the fragments, this is going to be disrupting the movement of potential animal dispersers, so frugivores. And so what you could expect and what we observe really is that most of the species that are found in these isolated patches are wind dispersed. Now, what we found here too is this indirect effect of low or the quality of the matrix. So these isolated patches, on top of having this like high distance between them, they also have a quality of the matrix that was very low. And the problem with this is that most of that area that was surrounding these fragments is mostly composed of tea plantations. On top of that, we found that there was an indirect effect of fragment area on the distance to edge and shape complexity, as we found also with functional richness. But in this case, we didn't find an effect of distance to edge to functional eveness. And then for functional divergence, we didn't find any effects of fragmentation on landscape level processes. So if we go back to this, what we're looking is there is a replacement of disturbance intolerant species replacing these disturbance tolerant species. So the fragments that still remain in this area are becoming more similar. And what we could expect is that in the future, they're going to be impacting the functioning and stability of that ecosystem. So this is just some of the fragments that we were looking at. And on top of this, we found that direct effect or that strong evidence that indirect and direct effects were affecting the functional diversity. And so this highlights the need of future studies to include both like the indirect and direct effects. to gain a more realistic understanding of the conservation status of the ecosystem, but also to better inform management efforts that are occurring in these areas. So what is next? The idea is to expand that multi-scale approach, but this would be conducted at Los Tuxtlas Forest, which is located in the state of Veracruz in southern Mexico. That's where I did my PhD dissertation, but over there I was looking at the population dynamics. So if you're interested, I can talk about that later. So this forest, it represents the northern edge of the distribution of neotropical forest, and is a very interesting forest because it combines elements from the north and the south. But the problem is that only 16% of the original forest, it's persisting this archipelago of forest fragments that you observe here. And this is because of the conversion of the continuous forest into pasture for cattle ranching. So right now I'm currently working on the proposal in collaboration with, again, Noel Beckman from Utah State University and Carol Garzón López from Universidad de los Andes. And the idea with this project is to compare what's happening in a continuous forest and forest fragments, and also see whether there is a functional homogenization of, or whether some functions are being lost due to forest fragmentation. On top of that, we want to see these spatial scale effects. So the problem with current fragmentation level studies is that they're looking at the plot or fragment level. So we cannot really assess the scale dependency of the fragmentation processes or even the effects of habitat loss. And also we cannot capture processes that are occurring at a larger scale. So the idea with this question is to take data at the plot, fragment, and also at the landscape level, and using a hierarchical model where we can also include the interaction of those processes to observe whether we obtain similar conclusions if we're looking at the plot, fragment, and landscape level, or we get in different conclusions of how habitat loss and fragmentation is affecting functional diversity. And then this is the most complicated one, because really what we're trying to see is there is a functional differentiation between the continuous forest and forest fragments that is happening over time. So really, the temporal changes or evidence of temporal changes on functional diversity is practically non-existent for fragmented landscapes. And so what we're trying to see is, using remote sensing, we're going to reconstruct the fragmentation process and also see the habitat loss, the amount of habitat that still remains. And then with that, with aerial imagery and also the individual delineation methods, what we're trying to see is if we can define some of the functional traits at the canopy level. So one example would be just the total canopy area, and see if those traits have varied over time between the continuous forest and forest fragments. So this would be one of the first studies to look at these multi-scale approaches in fragmentation. And this is something that really hasn't been explored, but is necessary to obtain a better understanding of the conservation status of this forest. In addition, we're trying to include information of traits that haven't been explored in fragmentation landscapes, or fragmented landscapes. So for example, at the root level, so we've been talking about this today, but at the root level, we're trying to see how that the species are responding to these changes that are occurring in the edge habitat where you have a reduction of water, right? So these water limitation responses. And that hasn't been really explored at the fragmentation level. And also, we're trying to include dispersal traits to look at how this isolation might not be only impacted the colonization ability, but also how it's going to be impacting those species interactions, so pollination, seed dispersal. That really hasn't been explored. So with this, I want to say thanks to the funding sources. So I put Tropenbos, which was essential for my undergraduate level work, where I started to work with the Janssen and Conner hypothesis in the Amazon, and that started to roll the ball, right, in terms of plant ecology. NSF, that was a part of my postdoc. Roofer, that allowed me to explore these effects of forest fragmentation in plant communities. And then forest geo, which I didn't have the time today, but we're working on a project on the plot nearby with Monique, that is a new postdoc. The collaborators, as we mentioned, Nathan Swenson from the University of Maryland, Bill Fagan, that was also my postdoc mentor, and Maria Natalia Umaña, that is also a strong collaborator. Yeah, with this, thank you so much, and let's stay tuned for the results for the forest fragmentation in the future.
Curious about the effect of soils on some of these phenomena that you're talking about, particularly the fragmentation, or a soil might make some species more vulnerable than others, or perhaps more robust.
Yes.
Could you comment about this?
Yeah, definitely. So the idea with the roots is also looking at those soil plant feedback. So what we're expecting is that the soil moisture is going to be lower in these edge habitats, right? So you could think about these edge habitats being filters. So what I'm trying to see is not only like this decrease in temperature, but also soil moisture, and how that's affecting the survival and growth of these species, and also the traits that are being selected for that. So definitely I'm going to be including the soil, and even fertility too, because the amount of nutrients is going to be different too. Mm-hmm.
So I guess what we might, something that I'm thinking of is looking for some sort of intensity of the functional trait value that would indicate whether or not that species experiences more negative density dependence or less negative density dependence.
Yeah, definitely. I think the next step of that project is looking at the species levels or species-specific responses, right? And the problem with the Janssen and Connell is that it has been conducted at the population level, but at the community level, there is no strong evidence, right? So we're still missing those. Yes, we know that some species are affected more strongly by those effects, and some species are not. I mean, we see some facilitation happening too. But it's very difficult. I think looking at, again, plant defense, it requires some type of experiments. This transcriptomic data can give you some information, but really it's just one step. We really don't know if those functions are being expressed too, right? So you will need to have an experiment to determine, yes, we have these functions, we describe these homologous genes, but are they expressing or not, right? So this is just like a stepping stone, right, on understanding the Janssen and Connell effects at the community level, but really is we still don't have really strong evidence of those effects at the community level. But I agree. I mean, you see these species, some species being erased because of these Janssen and Connell effects. But then the problem, and as I was mentioning before, so there's been this debate lately on how we are measuring a kind of specific density dependence, right? And this is just in terms of the abundance, it's a really mess right now that we're trying to understand. And also how we can better or propose better measurements of this type of effects. So the idea or the question is whether these Janssen and Connell effects are really strong in these plant communities, and whether also they are stronger in the temperate or tropical forests, as it has been suggested, or really, if they're not, they don't have those strong effects to structure these forest systems. So that's a question.
So I might have missed something. These are a lot of
Yeah, so some of those invasive species that you might have in mind, the stress tolerator, right? So they're gonna do very good on those type of habitats. So some of these traits might tell you whether, I mean, light demanding species that are also invasive species, and they tend to do very good in these habitats. So the idea with this is not only looking at the functional diversity, and this is just all the species, but also kind of like disentangle which are the species that are being selected. Also, what are the traits that are also being selected at the species level? So do you have these invasive species that are doing very good, right? They have like higher abundance in these areas. And maybe also the traits that we're finding is that they're correlated with this ability of colonizing by those invasive species. So right now the idea is just looking at the functional diversity, but then getting to the detail of the species level and see if it's just a light demanding species or pioneer species, or is a invasive species, and how that might be affecting those invasions, the responses in the future.
So like in your Tanzania, from your project from Tanzania, do you have any idea for how long those fragments are separated from like the main fragment?
Yeah, so there is the idea that has been for 100 years. And there is like some management efforts to decrease that level of like tea plantations. But I mean, it's been for a long time. It's one of those long-term experience or studies like the one in Brazil too, where it has like more than 100 years now. But the problem is that, I mean, we have that information, the fragmentation occur, but we cannot go back and take the measurements of functional diversity, so that evidence is non-existent. Yeah. Yeah, and so the I think 100 years is pretty long, but the problem in Los Tuxtlas, not a problem, but it's an effect that has happened 60, 70 years. So it's more recent than what I observed in Tanzania. And yes, you can have a mix of, especially in the large fragments, right? So you can have a mix of late successional pioneer species because they were just there. But I think now we're observing these drought events that are more severe, more frequent, and we can make the association with fragmentation and see like these effects on functional diversity. But yeah, I mean, I wish we had data, you know, in terms of like going back in Brazil or Tanzania and having that information of the function traits.
Okay, well, okay, so let's all thank to Jenny.
Thanks.